Welcome back to my YouTube channel. Let's do the part B here. Hope you have seen the part A in previous videos. Let's move to part B, which will carry 2 marks each. There will be 5 questions. Let's grab that 10 marks together. Here there is a diagram. You have to identify what it is. That is the A part. Identify and name the human embryonic stage. That is blastocyst. Blastocyst, what does it do? Mention its exact location, and we have to tell what is X, what is Y, and its function. This blastocyst will be getting attached or will be present in uterus or attached to endometrium of the uterus or the inner wall of the uterus. That is the answer for part B. And what is the answer for part C? What is X and Y? This is trophoblast or outer layer, and this Y is inner cell mass. Trophoblast will help the blastocyst to get attached to the uterus, and inner cell mass will help. It will differentiate into embryo in future. So answer is given here. Take a screenshot. From the next question, how the pollen tube is entering into the ovule. That is the question. See, when the pollen tube is forming, there will be 2 male gametes. This will enter into the ovule by the micropylar end. These are the keywords: micropylar end, through the synagogues guided by filiform apparatus. So there will be 3 keywords in this question. How pollen tube is entering into the ovule. Micropylar end, that is not the chelazal end, it's the micropylar end, through the synagogues, and it will be having filiform apparatus to guide the entry of pollen grain. And I told you there will be 2 gametes in one pollen tube. One will be going to the center and fuse with the polar nuclei, 2 polar nuclei, to form primary endosperm nucleus. That we call it as triple fusion. And one is remaining, that will go and fuse with egg, and that forms zygote. That we call it as syngamy. This is called a double fertilization. So what is double fertilization? You can write the same answer, that this big answer as well. That can be also asked. Even they ask the state the fate of male nuclei present in the pollen grain. What is double fertilization? Everything comes as this particular answer. So learn it properly. What is triple fusion? What is synchrony? What with which thing getting fused to form triple fusion and synchrony? Learn it properly and write down. That will be the perfect answer. There are 2 structures A and B. You have to tell what it is. Yeah, this A is plasmid or circular DNA. This is bacteriophage. What does it do? What is its importance? They are vectors helping to transfer the desired DNA to host. So what are their importance? They have copy number. They help us to do the transfer the host DNA, desired DNA to the host. Let's see the answer. See, plasmid. Write down anything, any answer you can write. It help us to carry the foreign DNA. As I told, it has selectable marker, it has high copy number. At least these 2 things you can remember. It has selectable marker and it has high copy number. And high copy number can be written in the bacteriophage as well. It has ability to replicate in bacterial cell. Oh, the plasmid or bacteriophage, whatever it may be. It has copy number or simply. And in the B part, they were asking, explain the process by which a bacterial cell can be made competent to take up foreign DNA from its surroundings. See, how can we make a cell competent? That is the question simply. Let's see it. So for it, we have to treat the bacterial cell with a specific concentration of calcium ion, divalent calcium. It increases efficiency where DNA enters the bacterial through the pores. So it helps the DNA to enter to the cell through the pores. And after that, we have to put them in ice bath. Once we put them in ice bath, then we put them in 42 degrees Celsius heat shock, then again to ice bath. So remember, first calcium ion, which will increase the efficiency by which the DNA is entering into the cell, then incubate in the ice bath, then 42 degrees Celsius, then again ice bath. Or if you remember these 4 steps, the competency, making competency to a cell is ready. See, what are the limitations of ecological pyramids? It does not take any account of one species remaining into two trophic level. It does not tell anything about it. And not only that, it only tell about food chain, not the food web. It does not say anything regarding food web. And there is no scope or inclusion of saprophytes. Fine. See, India is the 7th largest country in the world, and in India, 2.4 percentage of land area is there. That much of percentage of land area we have compared to the whole world. Remember that number, okay? And mention then what makes India one of the 12 mega biodiversity countries in the world. That is because 8.51 percentage share of the global species diversity. 8.1 percentage global species diversity is present in India. And this richness in this global species diversity or richness in flora and flora helping us to get included as the mega diversity country, including this endemic species richness. And what is endemic species? They are the species which is present in one particular area, one plant or animal present in India we can't see anywhere in the world, only in India. That is endemic species. Let's just like if you are present in your class or 12A, in the sense you are a student in 12A, we can't see you in commerce department or humanities department, right? That in the same way it is present in only in that area, in only that attendance register. The same way you are endemic to your class, the same way endemic means it is present only in that particular area. So we have completed the part B. Let's move to part C in next video. Stay tuned for it. Please give me a thumbs up if you like it.